This is for our final exam items, PowerPoint 3. And this is our final unit on medical micro. And again, folks, I'm hoping this information will still be relatively fresh in your mind. This was the last unit before our online lecture exam 3. And then do remember, folks, that on our comprehensive final exam, we will have the immunology, new information on non-specific defenses and specific acquired adaptive immunity. Excuse me. Okay, so we'll see if we can't get through this efficiently. Okay. All right, so medical micro introduction, so how to be a good pathogen. And folks, you'll recall there was 2 sections to this. The first section was on virulence factors, and the kind of subtitle was how to be a good pathogen. And then part 2 was the epidemiology, where we looked into reservoirs and transmission. So first of all, folks, remember the germ theory of disease states a germ, a microbe, can invade another organism and cause harm. And the germ theory of disease had to be proven. A lot of times people thought just spontaneously, like if you were a bad person, a microbial pathogen could arise spontaneously inside of you. So it was really important for folks to prove that the pathogens came from outside of us. We were invaded, right? And that was what caused disease. So Louis Pasteur and Robert Koch were in a competition to prove the germ theory of disease. Koch's lab won, if we could call it that. Koch's lab was the one that developed the use of agar to solidify media. And this was really important because very few microbes have the enzymes to digest agar. So when you're growing your microbes, since the agar isn't hydrolyzed, isn't digested, it remains solid. And also the agar will remain solid at relatively high temperatures. So Koch's lab, in using agar and agar plates, can guarantee they were working with pure cultures, which is essential to prove the germ theory of disease. Pasteur's lab was only working with broths, and thus they could never absolutely guarantee they had pure cultures. Okay, so then Koch developed 4 postulates to prove a specific microbe causes a specific infectious disease. So to prove that germ X cause infectious disease X, you have to fulfill these 4 steps or 4 postulates. So first of all, in all cases of the infectious disease X, you must isolate the suspect pathogen X. And very importantly, you have to grow it in pure culture. Okay, so you can see where pure culture is so important here. So this is our pure culture of pathogen X. And then the third step is to take healthy animals and inject them with your suspect pathogen X. And then the healthy animals have to develop the disease, and furthermore, you have to re-isolate pathogen X from the sick infected animals to prove that pathogen X causes infectious disease X. Now, not all pathogens can be grown in pure culture. So, for example, viruses, you have to grow them in another host cell. So with viruses, they've had some modifications, but generally, these steps, in many cases, help one prove a specific microbe causes a specific infectious disease. Do you remember, you guys, the inverse relationship between infectious dose 50 and LD50 and virulence? So the lower the infectious dose 50 or LD50, the higher the virulence. So in being a good pathogen, remember, folks, as we discussed with viruses, the first step in them being able to cause harm is the viruses must be able to attach to the host cell, and usually that's done with microbial adhesins that bind to specific host cell surface receptors. So many pathogens will have adhesins to bind to specific host cells. And remember, you guys, neutralizing antibodies can bind to the adhesins and then block attachment to the host cells, and that will block infection. Biofilms are another way that microbes can attach to surfaces within the host. So you'll recall that foreign objects in our bodies are great places for biofilms to form. Damaged tissues are great places for biofilms to form. And we want to remember the bacteria, they would secrete that sticky slime layer that lets them bind to these artificial surfaces or damaged tissues. They can grow, the biofilm gets thicker. And we want to remember that the bacteria are often much harder to kill when they're living in biofilms, are often in the stationary phase of growth, naturally more resistant to antibiotics. The layers of the biofilm would protect them from antibiotics, phagocytes, and antibodies. So we just want to remember that if you have a patient that is infected with a bacterial pathogen and they're living in biofilms, it's going to be much harder to kill them than if the bacteria, or the pathogen rather, was free floating or planktonic. So once you've bound, attached to your host, then you have to evade the host defenses. So you can camouflage yourself, you could coat yourself in host proteins, you could form the biofilms right, which will protect you from phagocytes and antibodies. You might synthesize an antiphagocytic capsule. You could make toxins that would kill the protective white blood cells sent to kill you. So we'll talk about leukocytes. Oops, let me back up here, you guys. So, all right, so this is just kind of hitting some points. So you guys remember leukocytes are toxins that pyogenic bacteria can produce to kill the white blood cells that arrive. So remember, a pyogen is something that makes pus, and we usually associate pyogenic bacteria with production of leukocytes. Pyrogenic means they're going to trigger fever, okay? And let's see here. So it would be good, folks, to review kind of the steps in Staph aureus invasion. So remember, you guys, that when Staph aureus first invades us, Staph aureus can make a hyaluronidase, so-called spreading factor, that digests the intracellular cement that holds our cells together, and so that lets the Staph aureus slip between our cells to invade deeper and deeper. When they're in low numbers, they'll make coagulase to form a protective fibrin clot, so they can live within that little protective fibrin fort. But then the problem is when they replicate to really high numbers, conditions within that little fibrin fort, that little abscess, are going to get wouldn't promote the survival of the Staph aureus, right? So there would be low nutrients, a lot of metabolic waste products. So using quorum sensing to sense their population size, the Staph aureus can turn off the genes for coagulase expression and turn on the genes for staphylokinase production. And the staphylokinase will break down the fibrin fort. And now that the Staph aureus are in high numbers, they can overwhelm those waiting phagocytes. We made a big deal, you guys, about toxins that bacteria can make. So remember the protein exotoxins that bacteria can make, both gram-positive and gram-negatives. So some of the vocabulary we need to remember are toxoids, are modified toxins which can cause no harm, yet they can trigger a protective antibody response. And these toxoids can be used as vaccines. For example, the DTaP vaccine protects against diphtheria, tetanus, and pertussis. Antitoxin are antibodies against the exotoxin. So, for example, antibodies could bind to the exotoxin, neutralize the toxin so the toxin can't bind to our cells. And if the exotoxins can't bind to our cells, they can't cause harm. So this table, you guys, we made kind of a big deal about it. So comparing exotoxins to endotoxins. Exotoxins are proteins, so a bacterium simply has to have the gene for the protein exotoxin, so gram-positive and gram-negatives can make it. Endotoxins, since endotoxins are the lipid A of lipopolysaccharide, we know only gram-negative bacteria can make them. Composition, so exotoxins are proteins, endotoxins lipid A of lipopolysaccharide. The location, usually but not always, exotoxins will be secreted from the living bacteria. There's always exceptions. And then endotoxins, since they're part of the bacteria's cell wall, usually large amounts aren't released until the bacteria die in lice and the cell wall starts to fall apart. Exotoxins are highly toxic, meaning tiny amounts like nanogram amounts can be really harmful, whereas endotoxins, they usually have lower toxicity. It means it takes more endotoxin to cause harm, but very often in so-called endotoxic shock, large numbers of gram-negative bacteria are lysing and dying, and so you can get massive release of endotoxin, which can be fatal. So signs and symptoms, usually exotoxins are kind of tissue or cell-specific, so there'll be variable effects. As a class, they're not pyrogenic, they won't trigger fevers. The endotoxins, however, have kind of classic signs and symptoms, so they are pyrogens, they'll trigger fever. You'll remember they trigger that cytokine cascade or cytokine storm. So because our blood vessels dilate and become leaky, we have hypotension, that decreases blood flow to organs. The endotoxin, the cytokine storm, can trigger disseminated intravascular coagulation, so we're starting to throw clots everywhere. We can progress to multiple organ system failure. If DIC goes on long enough, you use up all your clotting factors, and then you end up hemorrhaging out. So bottom line is the blood flow to cells and tissues is inadequate. So again, you go into multiple organ system failure, into shock and death. And this is often described as gram-negative sepsis or septic shock. Exotoxins, since they're proteins, they're sensitive to heat. They can be denatured. One exception is the Staph aureus enterotoxin. You might remember it's relatively stable with heating, right? And then in contrast, you guys, to the heat-sensitive exotoxins, endotoxins are unfortunately very heat-stable, resistant. You could autoclave endotoxin, like you could autoclave gram-negative bacteria, and you would kill the bacteria, but the endotoxins would still be active. And then this is important, you guys. So exotoxins, we can make toxoids of them. We can make antitoxin, and we can't make toxoids nor antitoxins to the endotoxins. Lipids tend to be really poor antigens. And it would be good for you guys to review the different types of botulism. So remember, botulism caused by the bacterium Clostridium botulinum, which makes endospores. There's a foodborne botulism in which Clostridium botulinum makes botulinum toxin in poorly canned foods, and then we ingest the botulinum toxin. So that's the foodborne intoxication, not an infection. But then sadly in children, little babies, maybe under 18 months of age, if we feed them honey, which is a rich source of Clostridium botulinum endospores, the endospores germinate in the baby's anaerobic intestine. The Clostridium botulinum grow and release botulinum toxin, so that's truly an infection. We end up with infant botulism. We can also have wound botulism, especially in folks that are IV drug users and don't have access to good sterile needles and syringes. They'll have a lot of abscesses, a lot of anaerobic tissue, and if they're injecting using literally a dirty needle that has botulinum endospores, the endospores can germinate in that anaerobic tissue, and you'll end up with wound botulism. Okay, and so with botulism, you might recall the botulinum toxin prevents the release of acetylcholine at the neuromuscular junction. So you get this flaccid, kind of ragdoll paralysis, and you end up dying from respiratory failure. And then folks do remember the cousin of Clostridium botulinum, Clostridium tetani. Clostridium tetani also makes endospores. So if the endospores get into anaerobic tissues like the umbilical stump of a baby, they'll germinate, release a tetanus toxin, which inhibits the release of inhibitory neurotransmitters. So you get this spastic paralysis that can also cause respiratory paralysis. So neonatal tetanus, and then the classic wound tetanus, like the old, stepping on the rusty nail. Antitoxin against a botulinum toxin, and antitoxin against a tetanus toxin could be given to these victims, these babies or an adult, to help preserve their life. This was just a cartoon, you guys, of kind of the sepsis. This is the cytokine storm that's caused by bacterial products, and the classic one, again, you guys, was endotoxin, the lipid A of lipopolysaccharide. And then, folks, just really briefly, we'll go to the epidemiology, the reservoirs and transmission. So you'll recall, folks, for infectious disease occurred, actually it's an interaction of three factors, so the host and environment and the microbial factors. So we talk about, if all the conditions are just right, you get the perfect storm, and that's when infectious disease occurs. And remembering that symbiosis is different organisms living together, right? And we said there's three different types of symbiosis. Win-win is mutualism. Win-neutral, one partner wins, the other is neither harm nor benefits. That's commensalism. And then parasitism, this is when one partner wins and the other partner is harmed. So when we talk about infectious disease agents, pathogens, we're really talking about parasitism. And the thing we wanted to remember was, although many of our mutualistic and commensalistic, well, primarily mutualistic microbes, they're actually really helpful. They can help defend us against opportunistic pathogens. We want to remember that if these mutualistic or commensalistic microbes end up in the wrong place in our bodies, or if we're severely immunocompromised, these symbiotic relationships can change. So a mutualist, if it's in the wrong place, can become a parasite. And again, just stressing you guys that the normal microbiome may become opportunistic pathogens if we become immunosuppressed. There's a change in the normal makeup of the microbiome. So, for example, if we take broad spectrum antibiotics, we kill all good protective lactic acid bacteria, then that permits the yeast Candida albicans to take over. And again, if the members of the normal microbiome are introduced into unusual sites, So for example, if we have an indwelling IV catheter or urinary catheter, microbes can be introduced into the bloodstream or into the bladder. Injuries, right? We maybe get skin microbes introduced into deeper tissues where they can cause true infections. How to be a good pathogen? We want to maintain a reservoir, and the three reservoirs are humans, animals, and remember, you guys, pathogens that can invade humans, which have non-human animal reservoirs, we call those zoonoses, and it's thought that most of the emerging new infectious diseases in the future will be zoonotic. And then the environment, if we have tough resistant pathogens, they can survive in water or soil. And again, folks, animal reservoirs, so these are pathogens that if they infect people, we'd call them zoonoses. So hantavirus with deer mice, bubonic plague, monkeypox with prairie dogs, rabies, right? Bats, skunks, dogs, I guess foxes here. And I put chronic wasting disease with a question mark because it's not been proven it could. Chronic wasting disease, the prion disease of deer, elk, and moose, it has not been shown to be zoonotic. But remember, you guys, never trust a prion. Transmission, so how does the pathogen leave the reservoir and reach a new host? So we talked about vertical transmission from mom to baby. Often this is across the placenta, or it might be at the time of birth, or sadly sometimes even in breast milk, for example. HIV can be transmitted to babies in infectious doses in breast milk. Okay, and then horizontal transfer or transmission, contact vehicle and vectors. And we're kind of making a joke of the F's of transmission. You guys, do remember that inanimate objects contaminated with pathogens are called fomites, and they can act as a way to transmit pathogens from an infected host to a new host. These are just some of the pathogens that can cause vertical infections. They can cross the placenta, and usually that's devastating, right? Because the baby developing in utero and the newborn, their immune system isn't competent yet. And this was just some examples of transmission. You guys, contact, there's direct, indirect, and then some people put droplet in a third example of contact transmission. Airborne, waterborne, and foodborne. And then some people include additional vehicles like blood or drugs, and then vector transmission. Healthcare associated infections, in the old days, these were called nosocomial infections. Often these are really serious because if you're in a hospital, it might mean you're already hurt or you're ill. And very often, bacteria acquired in the hospital, often they're resistant to antibiotics. So we want to remember that washing your hands, as Ignat Semmelweis told us, that's one of the best ways we can protect our patients. Okay, folks, I think that's it then. So yeah, I'll be checking emails, and it's just so wild to think this wild and crazy semester is drawing to an end. And again, you guys, I just want to thank you so much for your patience, your good humor, your adaptability, you're resilient. You guys are gonna do really well in the future, and we can get through these crazy times, right? So I'll probably post just like a little video just to kind of say goodbye. Fall, we're going to be at SCC, we're going to be teaching all of our classes online. So I was hoping I'd get to see some of you in the fall, but it looks like it might be a while. But definitely, guys, stay in touch. Let me know if there's any way I can help you in the future. And again, thank you. You guys have just been awesome, amazing.